This Week in Virology, the podcast about viruses, the kind that make you sick.
Hello, Ann Arbor.
Hello.
All right, this is great. It's time for This Week in Virology. This is episode 360, and today is October 23rd, 2015. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. I am in Ann Arbor, Michigan today, and if you listen to TWiM, you know I was here yesterday as well. We are celebrating the naming of the Department of Microbiology and Immunology as a Milestones in Microbiology site by the American Society for Microbiology. That means it's a historical site and has contributed a great deal to the field of microbiology. And yesterday on TWiM, we talked with members of this department who work on bacteria, and we talked about the history of the department. And today we're going to focus on faculty members who work on viruses. And I'm really pleased to have joining me today, I usually say from Southeastern Michigan, but I'm right here with her, Kathy Spindler.
Hi, everybody.
And Kathy is from right here in this Department of Microbiology and Immunology. The cool thing is yesterday we had Michelle Swanson, our TWiM co-host, and Elio Schechter, who's watching us do TWiV. Hey, Elio. Also joining me are three faculty members from this department. Right next to me, he is an associate professor in the department, Akira Ono. Welcome to TWiV.
Thanks for having me.
When I invited him by email, he responded yes, and he said, my palms are already sweating.
Right now, indeed.
No. Make him feel at home. He'll be fine, right? Okay. Don't be nervous.
Okay.
No one knows you and your work better than you.
Oh, really?
That's what I always...
That's disappointing.
Unfortunately, that's the way the science world goes. Next to Akira, he is an assistant professor here who I know quite well, Adam Loring.
Hi, thanks. Great to be here.
Welcome to TWiV. And finally, over there next to Kathy, he's a professor here in the department. I also know him quite well, Mike Imperiale.
Morning, Vincent.
Elio, Imperiale, right? Imperiale, but you don't say it that way.
No, it's been Americanized.
I know. So these are three virology faculty. We're going to talk about a little bit about the history of the department and their work. But first, what do we need to do first?
Talk about the weather.
Are you on with
I'm
the weather?
Set. So here in Southeastern Michigan, it's 44 degrees. Feels like 41 degrees. Humidity 83%, 6 miles per hour, wind from the east.
Nice. So now yesterday was quite warm here. It was a balmy fall day, and now it's getting chilly. Fall has set in. All right, that's the way it goes. It is late October. The other thing I have to tell you is that today is World Polio Day. And the WHO, Rotary International, who pays for a lot of polio eradication, they're having a big event today. They have a website called endpolio.org. Which you can go and check out all the things that are happening. There's a live stream coming out of New York tonight at 6:30 Eastern, where they're going to have people talking about the eradication effort. Time Magazine senior editor Jeff Kluger is going to moderate the discussion. They're going to have people like the UNICEF executive director, the CDC polio eradication branch chief, and then they have all these celebrities who are going to get up there and make people interested, because sometimes that's the only way you can get people interested. I know not you guys, not the listeners of TWiV, but so this is, the other thing is that the Gates Foundation, which pays for a lot of polio eradication, will triple any donations that are given, so this is a good cause. All right, so that's World Polio Day. And I just want to note that Adam and I both work on polio virus, and so we'll have to stop that one day, right? Do you think about that at all?
I do.
You got something lined up?
Every time I put in a grant.
Well, I'll tell you, there was a time when they first announced this eradication effort in 1980-something. When you wrote a polio grant then, right after it, the study section would say, why do we need to do this? It's going to be eradicated. And then after 20 years went by and it wasn't eradicated, they started leaving us alone. Right? So now I wonder, we're getting very close because 2 serotypes are gone, right? So
There are worse freezers than my -80 around, I know that.
mine is pretty bad. I have some old collections in it as well. So anyway. All right, so World Polio Day. Check that out. Kathy, what else is it today?
Today is also Mole Day. So it's October 23rd, and Mole Day is actually celebrated by moleday.org. So at 6:02 this morning, you could have thought about Avogadro, and at 6:02 tonight, you can think about Avogadro.
Well, when you said mole, I thought you meant the animal.
No.
No. Chemical mole.
So you can think about them.
Yeah. You
Yeah.
think about 10 to the 23rd animals. Yeah.
You guys like that? Is that cool?
Fun facts.
Of course, there are more viruses in the ocean than Avogadro's number, right? 10 to the 31st or 10 to the 30th, depending on who you talk to. All right, let's find out where these virologists have come from. Tell us, I know where you were born, of course, but tell us where you were born and educated and how you got here to Michigan.
Okay.
Akira.
So I was born in Tokyo, mostly grown up in Tokyo area. I had my 3-year-old to 5-year-old time. I grew in Queens, New York.
Wow.
So you might pick up my accent from New York. Not really. So
Yeah.
and
I know Queens. My wife was born in Queens. I used
is
to
that
visit
right?
all the time. You don't have a Queens accent, I'll tell you.
Queens English.
Mike was born not far from Queens. You're born in Brooklyn?
Brooklyn.
Does he have a Queens accent?
A little bit. I did.
Okay.
Yeah, so then for the undergrad, I did the sea urchin fertilization study and the effects of calcium pump. And that,
Tokyo, right?
in Tokyo. And then that led me to study the calcium homeostasis in grad school in University of Tokyo. And then in that lab, somehow I got into the virology. And that is because in that lab, we wanted to know what the calcium ATPase, calcium pump, is having any effect on cellular functions, and we don't know what cellular functions are affected by this. So we thought, okay, let's ask virus, because virus use all the cellular functions. So if the calcium ATPase inhibition inhibits virus, then we should find what step of the virus replication is inhibited by that. Then we essentially found that the transport is affected and quality control and so on. That was like a long time ago, and already Ari Helenius and so on found the quality control of influenza HA and so on. But that's the time when I was hooked to the virus-cell interaction, more from the standpoint of cell biology than virology. Some people say cell biology is a section of virology, so that's probably fine. Yeah.
That's what Ari would say.
Yeah, that's right.
Right.
Yeah, and then I did the short postdoc in NIID in Japan to learn the HIV technique after getting the PhD in University of Tokyo. Then moved to US to join Eric Free's lab in NIAID. I did the postdoc for 4 years and then 5 years, and I stayed there for 5 more years as a staff scientist. During that time, I really got lots of learning from Eric and working on the virus assembly of HIV, and I continue to work on that HIV assembly from cell-virus interaction standpoint.
So do you remember why you got interested in science to begin with?
Yeah, I guess probably the first encounter to the science is probably a TV program about dinosaur hunting, and And then I thought, okay, yes, I got to be those dinosaur hunter. And Ocean Marsh, Cope, and those people. But I realized at the time there is no dinosaur bone found in Japan, and in US you got lots of dinosaurs. It's so cool. But okay, then I should do something else other than dinosaur, and then gradually, okay, maybe mammals. then I found I'm not so good at handling mammals. And okay, maybe cell. And that's how I get into the cell virus interaction.
I have to recount, remember what Jens Kuhn said a couple of episodes ago? I asked him, do you have to isolate a virus to be able to classify it according to ICTV? And he said, well, you know, T-Rex is classified and we never had a living T-Rex. Right? It's a good point.
Right.
I love that. So one of the many things I like about Japan, I have to say something in Japanese, so my son, if he ever watches this, because he taught me this. It has nothing to do with us being here, but it's the only thing I know. Itadakimasu.
Great. Yeah.
Except we're not eating, so it's
that means let's eat.
The other thing I really like about Japan is their pens. Are these Japanese pens?
Yeah, of course.
Oh, these are
It's from Pilot, and
Thank you very much. Nice, medium and fine point. So
Vincent, I have my jet pen here.
I turned cat. So jetpens.com is an American company that sells all Japanese pens.
Oh, is that right?
And it's awesome. I have a fetish for pens for sure. There's a big store in Tokyo, one of those department stores, and they have a whole floor of pens.
Oh yeah.
What is the big department store called? Anyway, it's awesome.
Yeah.
Thank you very much for your pens. Adam, where are you from?
I was born in San Francisco on the Presidio. My dad was in the Army. But I grew up in Florida when my dad left the Army. From when I was 4 to when I went to college, I grew up in Florida. And then I was an undergraduate at Yale, and that's when I first worked in a lab and got interested in science. And I studied Rab GTPases in yeast. And I was very interested in signal transduction and oncogenes and things like that. And then after there, I went to the University of Washington in Seattle, did the MD PhD program there. And that's when I got into viruses, sort of by accident. And I worked with Julie Overbaugh, who's a former TWiV guest, and worked on feline leukemia virus, which at that time was still considered a model system for HIV and SIV, and studied the pathogenesis of variants that had evolved in vivo. And so that's how I started to get into viral evolution. And then after 8 years in Seattle, I went down to San Francisco, where I did my clinical training. So I did a residency and a fellowship in infectious diseases there at the University of California. And then did a postdoc also in San Francisco with Raul Andino, who I think has been on a couple times. And Raul's one of his interests is RNA virus evolution and using poliovirus. So that's when I started working on poliovirus in Raul's lab. And then after that, I came here. I've been here just over 3 years. Met Mike and Kathy and Christiana at a Gordon conference when I was starting my job search. And so the rest is either history or a terrible mistake on the department's part.
We will see.
We'll see.
We'll see.
The jury is out.
So I met you in Raul's lab when I visited.
Yeah.
Did a podcast with him.
Yeah.
So Raul was on the first TWiV we did in front of an audience.
Oh, wow.
It was like episode 30 or 40, and there was one person in the audience. It was at
Adam?
Yeah, it wasn't Adam. It was this was an ASM meeting, and she was from the National Academy of Sciences. She was really into science communication. She actually asked a question, And we had actually, Dixon had brought some books, and he said, anyone who asks a question will get a book. So she got a book. And then she asked another one. She said, can I have another book? But yeah, it's just now it's very different. We have, I don't know, 50 people here, and this is great. All right, Mike, what's your history?
So I grew up in Brooklyn, and then I went to Columbia University as an undergrad.
So did you grow up far from Tony Fauci's?
No, in fact,
Okay, you didn't know him at the time.
interesting.
Right?
I didn't know him, but my sister's ex-husband dated Tony Fauci's sister before he met my sister. So there's this kind of
Mike puts that on his biosked.
so 3 degrees of Tony Fauci.
Okay.
No, so I did not know Tony, although he grew up very close to where.
He had his family had a pharmacy called Fauci Pharmacy.
I didn't know that.
And I found a picture. So we interviewed him a couple of years ago, and I found a picture of Fauci Pharmacy on the web. It's long. It's quite old. Yeah. So there's no Imperiale Pharmacy.
There is not.
No, but I have found places in Rome that were Imperiali, and in Trieste too, and take pictures and
Send
show
it.
them.
Yeah, but probably not the family name, more the word imperial, right? So.
Okay, sorry.
So I went to Columbia, and I thought I was going to be a dentist because I had an uncle, Vinny, who was a dentist, and he wanted me to, join him in his practice, and looking forward to retiring. But then I started doing research. So I was working with Larry Chasen, doing somatic cell genetics, and I just got hooked, and I thought research was really cool, and so I decided to go to graduate school. And I was all set to go to Stony Brook, and then Columbia offered me a fellowship, which I thought was very, very cool. So I ended up staying at Columbia. And my original intent was to study gene regulation, because that's what I was really interested in. And for a variety of reasons, in between the time I decided to go to Columbia and started that fall, the labs that were doing gene regulation were not available. And so I had a choice, right, which was to sort of pick a different topic or transfer to somewhere else. And I decided to stay. And at that time, each faculty member would give a little blurb about their research. And I became very interested in a person who was studying immunology. And so my graduate work was in T-cell immunology. But then as a postdoc, I wanted to get back to studying gene regulation. And as many of you know, at that time, really the only models to study eukaryotic gene regulation were viruses. And so I moved across town to Rockefeller University and joined Joe Nevin's lab and started studying regulation of expression in adenovirus. And it was that work that got me the job here in Ann Arbor in 1984. And so I've been west of the Hudson River ever since.
I can't believe you grew up and got educated in New York and you're out here. I mean, not that there's anything bad about it, but it's very different, right?
It is.
And
it was a bit of a culture shock moving out here. But it's a great place. It's a great institution.
You still have your accent, right?
A little bit. Some people tell me I do, but I know of other people who have lived here from New York just as long who have a much stronger accent.
You certainly have your edge.
Thank you.
His edge. So Larry Chasen, he's still around, and I just was on a student committee with him, so he's still going strong. So you're at Columbia in the 80s or late 70s?
So undergrad in the mid 70s, and then graduate from 76 to 81.
Okay, all at Morningside, right?
Yes.
Okay, good. So that, we have great respect for Mike because of his Columbia background for sure. Now, we know all about Kathy because Kathy's on TWiV every week, but maybe you could tell us a little bit about virology at UM.
Sure. So we have this history talk yesterday by Paul Kazanjian, who's a chief or division chief of infectious disease in internal medicine. And he told us all about Frederick Novi and told us how Frederick Novi was probably the first to work on viruses here at the University of Michigan. And either Adam or you could expand on that a little bit, and then we'll come back to some more recent.
So he was the first chair of.
He was the first chair of the then Department of Bacteriology.
And this was the first bacteriology department in the country, correct?
I think that's correct. Yes. Although there may be some quibbles with the history of medicine people. I'm not sure about that.
All right. Do you know anything about his virus work?
No.
All right. I'll let you off the hook. So I looked this. Kathy emailed me this morning. And there's a paper on PubMed First of all, if you search for his name, Novi FG, you find papers going back to like 1890, but there's no text, it's just a citation. And this one is called the rat virus. It's his only virus paper, and it was published in Journal of Infectious Diseases in 1953. The authors were Novi, Perkins, Chambers, and Paul TWiV, author of, what was the name of that book? Microbe Hunter, which I read many years ago. And I wasn't able to get the text for this paper in my hotel, and here it asks me to pay for the article.
And
That's affected Novi's H-index.
I couldn't get it to print out on my printer in time to bring it to you. So.
There's no abstract, so I don't actually know what he did. So.
Well, so what Powell talked about yesterday was that he was injecting rats with material that had bacteria in it or not, and some of the material that didn't have bacteria in it still was killing the rats. And so it had been filtered, so that was his clue that it was a filterable agent. And at some point, an EM image was taken, and I don't know if it was back then or later, but what Powell described was that in his late 80s, they had the tools to figure out that this was a rat virus. So we'll all have to download it later using mGedit and check it out.
Well, we'll talk about it next time. I like the title, The Rat Virus. Right?
Yes, pretty definitive.
There's no more. I also like the year it was published, 1953.
Right.
And you guys, if you're TWiV fans, you should know why I like that year. Sorry, go ahead.
Right. So the other thing that we learned yesterday about Frederick Novi is that he was really probably the first physician who did basic science. Or at least that was the case, someone who did basic science here at the University of Michigan. So that was exciting. And just to finish off kind of the things from the talks that we heard yesterday, we also had President Mark Schlissel here, president of the University of Michigan. He's an immunologist, trained at the same place as Vincent for his postdoc at David Baltimore's lab. And he talked, and what we found exciting about that was that he really tied microbiology to molecular biology, and all of the molecular biology that then blossomed into all the kinds of biology that's being done today. So that was really nice to have those two things, learning about the foundations of basic science and also tying in all the virology or microbiology, much of which was virology again, to the history of science. And both of those eventually will be available as video recordings. And I'll probably pick them as a pick when that happens.
So Schlissel is 4 years younger than I am. Right? He's your president. This is really remarkable. Although he's not the youngest university president. Does anybody know who that was actually? The youngest university president ever?
What's his name?
He's the brother of a...
Botstein.
Yeah, he's Leon Botstein, David Botstein's brother. He was 26 years old when he became president of Bard College. He was a orchestral conductor, and he's still there. He's been there for ages, in his 40s now. It's amazing. And David Botstein, the yeast geneticist, is his brother. So Schlissel trained a few years after I did in the Baltimore lab. And yesterday on stage, I took a selfie of me and him together, and that's on Twitter if you want to see it. So check that out.
Okay, so after Frederick Novi, we kind of have some virologists that were in the department and some that were not. And Tommy Francis is one that we've talked about before. He was not in our department. He was at the School of Public Health. I don't know if you want to expound a little on Tommy Francis?
Yeah, so it's this great place to do flu and polio, and Tommy Francis is kind of the guy. He was, I think, the second person to isolate flu, and that was actually PR8, which is a strain. And I was telling Kathy beforehand, when I set up and wanted to start working with PR8, I actually had to fill out an MTA to get it. And I was like, well, we shouldn't have to do this. No. It actually came from us originally. And he did a lot. He formulated, I think it's him, is the whole model of original antigenic sin, dates back to work he did. He then was Jonas Salk's essentially postdoc mentor. and ran the Salk vaccine trial, which was it 60 years ago, was announced a couple blocks away
54.
at the Rackham Auditorium.
1955.
And I don't know how big, I mean, it's a remarkably large trial in terms of the number of people
It was
who were in it.
1.8 million.
Yeah. Yeah. Which is absolutely, I mean, think about that. Think of you can't even do a vaccine trial that big today.
Right.
So that's pretty remarkable. And then I don't know if you're going to mention John Masab as well. So also at the School of Public Health, who did a lot of great influenza work and developed the virus that became flu mist.
Right.
So
When I was a graduate student with Peter Palese, I came in the lab and he handed me these strains and he said, these are from Masab at Michigan, just see if you can do something with them. And they were cold adapted strains, and that's what became flu mist. Yeah. Cool.
I think it's called Ann Arbor,
Yeah,
you know,
it was the
CA.
tube was AA / something, yeah.
Yeah.
Cool, all right.
I don't know if you have others.
So Mike, you can talk a little about Mike Levine?
So Mike Levine was in the Department of Human Genetics, and really, in my view, was one of the first people to really start taking some molecular biology tools and applying them to mammalian viruses, and did a lot of the pioneering work on herpes simplex virus and some of it. And at the time when I came here, the university was really starting to build molecular biology. So Fred Neidhart in our department and Mike and some others were very interested in making Michigan a place where molecular biology was going to grow. And in fact, my recruitment was part of a specific effort targeted towards that. And so Bill Brockman, who was in our department and worked with SV40, had trained with Dan Nathans, was part of that. And so David Friedman was brought in, I think, as part of that early effort. And so really, we had sort of a nucleus building at that time of individuals who were studying viruses to a large degree as model systems for molecular biology. Again, coming back to this point I made before, which is at that time, they were really the only tractable systems to study mammalian genetics and such. So that's how this sort of department, grew its virology in the early days.
So a little bit more about Mike Levine. He also worked on P22, the bacteriophage, and one of his postdocs was Ham Smith of a restriction enzyme and Nobel Prize fame.
And Bothstein also trained with Mike Levine.
Oh, I didn't know that. Okay, all right. David told me that yesterday. I'd forgotten that. Yep. And then another person that trained with Mike Levine was Ethel Jackson, and she worked on P22, and so she was a faculty member in microbiology and immunology, so she's the first female member of our department, and she was working on viruses.
Cool.
So then we go to modern era. We've had connections with lots of people doing virology in other departments, particularly School of Public Health and internal medicine, infectious disease types. And then Mike and I worked out a chronology of everybody who's come since him. And anyway, it ends with Adam. So he's our most recent virology hire.
Again, they haven't done any since. So
In fact, Adam's on the back page, but.
Of course, the most important thing is all the virologists who have been on TWiV, right?
Right, so Kathy Collins was the first. She was on TWiV 133. I think you did that one with her by Skype.
Correct.
And then in November of 2011, so just about 4 years ago, Stefan Taubi, a postdoc in Christiana's lab, invited Vincent to give a seminar, and I invited Rich Condit to give a seminar. We had them come back to back with the sole intent of getting to do a live TWiV here. And so that happened, and Alice Telesnitsky and I were on that TWiV. And then Mike was on TWiV 182, I think by Skype. Not so much talking about his work, but more about the H5N1 and NSABB issues. And then Christiana Wobis was on at the TWiV in Penn State. That's TWiV number 243.
So it was shortly after we did our TWiV here 4 years ago that I said, do you want to join us, right?
And I said yes.
And you said yes. But it took her a long time to decide.
No, not really.
Okay, that's a little bit of virology history here. I can see why you would come.
Yeah,
You have a lot of good colleagues around,
it's a great place.
even though it's in the middle of nowhere. I like to bug people like that, right? Okay, let's talk a little bit about your science. This is what we do on TWiV often. We have 3 of you, so it's a challenge, but we can do it. Maybe we'll start with Mike. And I know you've worked on both adenoviruses and polyomaviruses most of your career, correct?
Correct.
I like to talk about polyomas. Is that okay? So how did you get, I think some of your postdoc training was, no, that was with Joe Nevins, right? So you wouldn't have done polyoma there. So where did that come from?
Yeah, so the polyoma sort of came through sort of an unfortunate incident, which is that Bill Brockman, who was the person who was primarily responsible for my recruitment here and studied SV40 biology, passed away a month before I arrived on campus. And so the department was trying to figure out how his students at that time were going to finish up. And they asked me if I would take over as PI on his R01 to allow his students to finish. And the intent would be that once they finished up, then the R01 would run out. That would be the end of it. But I became really interested in the project, and I decided to try to renew that R01, which I did. And so that's what sort of got us into the polyomavirus field. And then there was a student who joined the lab a few years later and said, Mike, why are we studying this monkey virus? Why aren't we studying a virus that causes human disease? And of course, we talked about it being a model system and what a great model it was, but I challenged her to go find a human polyomavirus that we should study. And she went through the literature and came back and said, why don't we do some work with BK virus? And that's how we got interested in BK virus. At that time, it was just beginning to become a significant pathogen in transplant patients with the advent of new immunosuppressive drugs that were allowing the virus to kind of rear its head. And that's how we got interested in the virus, and we've been studying it ever since.
What's that student's name?
Kimya Harris is the student who originally was doing that work.
So now how many human polyomaviruses are we aware of?
So there are 13 human polyomaviruses. Only 5 of them are known to cause any disease, however.
This is mainly in immunosuppressed people?
Mainly in immunosuppressed people.
And what fraction of the humans have polyomaviruses of one sort or another?
So pretty much almost everyone. So 90% of us are BKV positive, a little bit lower percentage JC. The other viruses, the serology is around in the 60 to 70% range. So yeah, we're all...
Are we viremic?
Generally not. So JC virus is the one that seems to be excreted the most. So the polyomaviruses live in 2 places, the urinary tract or on the skin. And JC is excreted into the urine pretty frequently, and then BK follows that. So we're not viremic, though. Generally, viremia is only present in kidney transplant patients who have BKV disease.
Okay, so most of us are uremic. Is that the right word?
Viruric.
It's viruria, yeah. Virus in the urine. All the time, or
not all the time, periodically.
And what makes it?
So that's unclear. We really don't know. First of all, we really don't know where in the urinary tract the virus persists, and then we don't know what controls persistence and reactivation. That's one of the things that our lab's really interested in trying to figure out.
I always tell my class in virology, I said every time you go into a urinal, it's for the men's room anyway, you've got polyomaviruses everywhere because people flush and it aerosolizes. Should I stop telling them that?
No, you probably shouldn't. Okay.
They get very scared at me. So everybody has some kind of polyomavirus. Could it be beneficial?
So that's one of the things that I think about, because why would the virus have evolved to persist like that if it didn't play some beneficial role? So you can imagine that it does similar things to the microbiome, where maybe it keeps the immune response sort of at some heightened level of awareness or something along those lines.
Yeah.
So sure.
Yeah, I'm thinking so maybe it makes a low level of interferon that could be protective or something like that. How would we figure out if it's beneficial or not?
I think it'd be pretty tough. So some people are talking about developing vaccines against some of the polyomaviruses that do cause disease in immunosuppressed individuals. And, I suppose, one side effect of those vaccines could be if there's some beneficial role that you might see that go away. But otherwise, you'd have to use animal models. And there really is only one good animal model, which is mouse polyomavirus, although The mouse polyomavirus is genetically slightly different than the human viruses. So it may not be the best model for understanding.
So do most wild mice have mouse polyomaviruses?
I don't know whether most wild mice have them or not. Kathy, do you know that, Kathy?
I don't know.
Yeah.
So at some point in the future, we're going to have broad spectrum antivirals, I would guess. And I think they're going to be wiping out a lot of our eukaryotic virome, and we'll see if that's a problem.
Yeah. Well, so the thing with polyomaviruses, right, is that they're almost completely dependent on the host cell machinery for their replication. So the only enzymatic function they encode is a large T antigen, is the helicase that starts to unwind the viral genome during DNA replication. But otherwise, it's completely cellular. So unless these broad spectrum antivirals affect some cellular functions, the polyomaviruses may be immune to that.
Right. I don't know what the target's going to be. There's a whole class that could hit just envelope viruses, and that obviously wouldn't do that. But for the non-enveloped, I don't know what it would be. But I tell my class that, and once a student came down and said, "Wouldn't that be a good way to figure out if viruses are beneficial or not?" I said, "Yeah, it could be."
Well, I think the other place we might learn something is that people now are trying to develop a next generation of papillomavirus vaccines that would target a much wider range of the 100 and whatever types of human papillomaviruses. And again, there are human papillomaviruses all over our skin. And so if they're playing some beneficial role and now you start vaccinating against pan-papillomaviruses, that potentially...
Yeah, we'll see. But there's no move to immunize against the other polyomaviruses, the 13 or so that you talked about, right?
No, there's not, because in terms of the percentage of people who get disease, I think there's no market really.
So, in a transplant patient, is there going to be some way to deal with disease caused by immunosuppression?
Potentially, if we could come up with new targets for antivirals, that would be one way. Or if we really could specifically jack up the immune response in those transplant patients, even though they are immunosuppressed somehow. Potentially by, say, vaccinate before you do the immunosuppression, there might be ways to...
So Adam, as an ID person, do you ever think about these sorts of issues?
Yeah, well, I do 2 weeks of service here and there over in the hospital. I just came off, which is probably why I'm sick. And I saw someone who had BK virus. Went and told Mike. Because particularly where I see it is in kidney transplant patients, and so this gentleman had lost his kidney graft because of BK virus, and then what you have to do is you have to reduce the immunosuppression, and so the body's immune system can fight it. But then if you reduce the immunosuppression, then you can lose your graft, you lose your transplanted organs. So it's a real problem, and so I think the best idea would probably be before someone gets transplanted, vaccinate and in the run up to transplant so that they can hopefully not have it as a problem, because then a lot of the drugs that are used for BK are renally toxic. They're not very good for the kidneys, so it's a real bind.
But that's only one of many viruses you have to worry about in a transplant patient.
CMV is a big
CMV, right, and others as well. There's also a brain disease caused in immunosuppressed patients by polyoma, right?
So that's progressive multifocal leukoencephalopathy,
wow,
which is caused by JC polyomavirus.
Right.
And so, that was one of the AIDS-defining illnesses in the early days of AIDS. But now with heart, PML caused by JC has really sort of, decreased. But where it's becoming more of a problem now is in patients who are receiving biologics, and in particular monoclonal antibodies that are targeted towards sort of tamping down the immune response in autoimmune diseases such as multiple sclerosis. And the interesting thing there, and sort of the dilemma, is that only a very small percentage of those patients are getting PML. And it's unclear what the factors are that are contributing to whether or not these patients are going to get PML or not. And so the drug companies that are developing these biologics are very interested in trying to sort out what can we predict which patients are most likely to get PML, and then we won't put them on this therapy.
Right. So Mike, recently you had a very interesting story on microRNAs and regulation of BK. Can you tell us that story? I think our listeners would like it.
Yeah, sure. So The story here was that we were doing experiments to try to understand the genetics of the virus. So the virus that circulates through the population is referred to as the archetype of virus, and it doesn't replicate in cell culture. And so pretty much everything we know about BKV is gleaned from experiments using rearranged variants that have differences in the region of the virus that controls expression of the viral genes and replication of the viral genome.
Is that because they rearrange in cell culture?
You can select for rearrangement in cell culture, but also you get these rearranged variants out of patients who have BKV disease. So there's some selection in these patients, presumably because you're getting so much replication for these rearrangements. And so there's a student in the lab who is interested in why this is. Why can't the archetype virus replicate? And so the first thing that occurs when the virus infects cells is that it expresses its early genes, and one of those early genes encodes a protein called large T antigen, which is the master regulator of infection. So it induces the cell to enter into S phase, which is when the DNA synthesis machinery the virus needs gets produced. And also T antigen is this helicase that binds to the viral origin of DNA replication and recruits the cellular DNA synthetic machinery. And so the first thing she asked was, well, does the archetype virus make any T antigen in cells in culture? And the answer was no. And so the obvious experiment is let's provide T antigen in trans. And so when we did that off of an expression vector, we were now able to boost up DNA replication. But when we co-transfected in the archetype genome and this rearranged genome that produces tons of T antigen, we got the opposite effect. So it's something from the archetype genome was shutting down replication of the rearranged genome. And so it turns out that the polyomaviruses encode a microRNA that is complementary to the T antigen messenger RNA. So when you make the microRNA, you don't make any T antigen. And so what we hypothesized was that what's happening in the archetype virus is you're making a lot of microRNA, and that's preventing T antigen expression, and thereby you're not getting any replication. And so what we showed was that if you mutate the microRNA so it can't be produced, now the archetype virus will replicate just fine. So polyomaviruses have a circular genome, so basically the microRNA would be transcribed in one direction and the messenger RNA would be transcribed in the other direction from bidirectional promoters in this regulatory region. And so I think the story that you want to hear is that we were asking how can we show this, and so my student Nicole Brokema said, well, let's just take those promoters and flip them. Because that should then basically turn the archetype into a rearranged and the rearranged into an archetype. And we were talking about this, and I'm thinking, God, but we don't know anything about these promoters. We don't know even what the Tata box says, anything like that. And finally, we just decided, it's an easy experiment. Let's just do it. And we did the experiment. In fact, we got the result that we predicted, which is that we were able to basically turn one virus into the other genetically by just doing this. And so the moral of the story, which I told at the ASV meeting a couple of years ago, is if an experiment is simple, just do it, and don't spend time talking yourself out of it.
So you did a gain of function experiment.
We did a gain of function experiment, exactly.
So these rearrangements, which lead to increased microRNA production occur?
No, the rearrangements lead to decreased microRNA and more T antigen.
In the archetypes?
Oh, yeah, in the archetype.
So you have more T antigen in the archetypes. Is that the
No,
outcome?
sorry. So basically, our model is that there's a balance between the microRNA and the T antigen. So in the archetype, you make more microRNA, so you don't make T antigen.
Right, and you said that these archetypes evolve in patients typically, right?
To persist.
So that's why this happens, to encourage persistence, right?
That's our model, yes.
And then the virus still has to spread, so how does it make virus to do that?
Yeah, so periodically there is replication, even in healthy individuals, of the archetype virus, and it gets excreted into urine. So that's one way the virus may spread. The other thing we don't know is what's the original route of transmission. So is it through the urine, or is there potentially some other route, such as a respiratory route, that we don't know about? And the reason for speculating about that is that most people seroconvert in very early childhood. And so epidemiologically, it sort of looks like a respiratory pathogen.
Right, well, certainly.
And you can detect the virus in salivary glands and in tonsillar tissue.
Okay. So there is enough virus production, even with low T antigen, to allow spread?
Presumably.
Is that what you're saying? So the microRNA doesn't shut it off completely, it just dampens it.
Or maybe, from time to time, something flips that switch so that you don't make enough microRNA. You get a little bit of replication. The immune system comes in, shuts it back down, resets the system.
We always seem to use these stochastic excuses to explain virus behavior, right? But
Exactly.
when we can't figure out what the triggers are, we say, well, it randomly just flips a switch.
Yeah.
So let's talk a little. You've been very vocal on, as Kathy mentioned before, the H5N1 experiments. You were on the NSABB years ago, right?
Yes.
You're no longer on that, right?
That's right.
And now, more recently, that has transformed into a discussion, debate about gain-of-function experiments, which, not the greatest term, but where you give a new property to a virus. And some individuals are concerned about that. And right now we have a moratorium on certain kinds of gain-of-function experiments. So you've written a lot. If you go search your PubMed, there's a bunch of BK papers most recently, and then there's 2 pages of gain-of-function, mostly with Arturo Casadeval, right? So why are you doing this, and what's your position on the whole situation?
So I'm doing this because I think it's important, because I think that there's a concern here that we're not going to be able to do the science that's needed to really make a difference in terms of some of these important diseases, such as avian influenza or MERS or SARS and that. And there are some people out there who really just want to shut down that entire category of experimentation. Because of concerns that if one of these viruses happens to make its way out of the lab and it really has the properties that are predicted by these laboratory experiments, that could be a huge disaster. So where I come at it is that I think there's been a lot of sort of hysteria, and I think that we really have to take a step back and think about what are the important questions that we're trying to address. Can we address them in a safe manner? And then can we make the argument to the public and to those who are regulating this type of experimentation that there is enough of a benefit that we have to be able to accept some low level of risk. And the problem, right, is that it's really hard to quantify either of those sides of the equation. And so, and that's just in terms of, sort of the practical applications. And, as we've discussed before, there's also just the sort of the basic accumulation of knowledge, right, that in and of itself is an important thing to be thinking about. And so there are all these variables flying around there. There's some very, very heated debates. And what Arturo and I really wanted to set out to do is to try to maybe, put a little bit more of a, fact-based, get some facts on the table about, what we know, what we don't know, what's real and what's not, what's doable and what's not, and then try to frame the discussion in that manner. So one of the things that we've argued is that the way the discussion has been framed in terms of the term dual use has really done a disservice, because just using the term dual use to apply to beneficial research that potentially could be misused puts the idea that misuse is maybe one of the major things that can happen, and I think hopefully we would all agree that the chances of that are very, very small.
So I get the sense that you're not a proponent of the moratorium.
I'm not a proponent of the moratorium. I think that there are ways to accomplish that without this sort of blanket type of stop. I think that that what they should be doing, and presumably they have been doing since the H5N1 discussions back in 2013, is that they're supposed to be looking at each particular experiment, right, and asking, is there a risk here or not, and to sort of just say we're going to take everything that's being done with these agents and stop it. Potentially is going to hurt us than do more harm.
Do you have a sense what's going to happen with the current moratorium? When is it going to be over? What's going to be the outcome?
Yeah, I don't have a good sense of when it's going to be over. I think what I had heard was the original intent was that it was going to be about a year, but we're a year in now,
right.
and we're nowhere close to that. I know that the government has contracted out to a company the idea of coming up with a way to develop a risk-benefit analysis, and they've been looking into that, interviewing various people. I don't know if they spoke to you. I spoke with them on the phone a few months ago. And then they're going to make recommendations to the NSABB, and then the NSABB is going to make recommendations to the government, and then I don't know, where it's going to go from there. I could tell you from my experience on NSABB that government policy making is a very slow process. So I don't know what they're going to do about this moratorium. But the thing is, right, this only applies to U.S. government-funded work. And so the work, is going to take place in other countries. And that's another concern, which is whether, we in the U.S. are willing to cede our ability to do this work to other
Unfortunately, among other experiments, our understanding of influenza transmission is done largely in 2 labs that are NIH funded, and that work can't go on at the moment. I think that's unfortunate because, as you said, this is important. We need to know the answers to this. Do you have a sense, I have a feeling that this whole moratorium came about from people outside the field who got frightened by a lot of the rhetoric used by certain individuals, and the virology community itself is not as concerned. Is that your sense, or do you think there are virologists who are also concerned?
Well, I think there are virologists who are concerned. There's Simon Wayne Hobson has been one of the big proponents of not doing these types of experiments. But I think if you look at the virology field in general, I think most people are apathetic, and then there's a larger group of people who are not as concerned as those who are concerned.
Yeah. Are you guys apathetic?
Sort of.
Well, it could spread to encompass everybody's virus or even bacterial work, right? And once this is a slippery slope. Once you start making regulations about what you can do, it's very easy to extend those and go farther and farther. So that's why we all need to really be part of this debate, and I always talk about it.
Well, and that already happened with the moratorium, because extending the moratorium to MERS and SARS, which are already transmissible from human to human, I don't think anybody in their right mind would want to make them more transmissible. So there was
there's certainly no point in doing that, right? Because they are already.
So fortunately, they backed off on some of that.
Yeah. Well, I think as a community, we have to be very vocal. You have to get involved. You have to look at what's going on. You can't just leave it to Mike and Arturo and a few others to do this, right? Okay, let's move on to Adam. Let's talk a little bit about, you're interested in viral evolution.
Yeah.
Can I start by asking you to tell me what is a quasi-species?
Yay!
Now I'm going to get in trouble.
No trouble. I just want to know your take.
Adam's definition of a quasi-species.
Yeah. So as I've gone further, I've made a deliberate effort to not use the term.
Okay.
And because it's one of those terms that means different things to different people, and it becomes overly jargonized, I think, and so then it can distort what you're actually meaning. So I'll explain. So a quasi-species, well, it dates back to this. I mean, it gets a couple lines of evidence. So Manfred Eigen, who I think was a chemist, became interested in understanding the idea of what would happen in a situation where you had kind of self-replicating entity that was highly error-prone and that could lead to large numbers of progeny, large population size, and what did that mean for evolution? And so a very kind of theoretical approach to this. And then so he developed this theory called quasi-species theory that would explain, how evolution would behave. And the idea was that if you have an entity that makes a lot of mistakes or mutations when it replicates and makes large population sizes, that you essentially evolution would function perhaps differently, and natural selection would act on the group, the entire entity, this quasi-species, rather than on individual, entities within the population. And so that's the idea. And so, very beautiful theoretical work, discussing this that I'll plead. I took calculus last 30 years ago, probably. So, I can't pretend that I follow it. And so, but then, in the 70s, Esteban Domingo and John Holland and others working with RNA phages, and then Domingo with FMDV, foot and mouth disease virus, we're like, well, gee, these viruses do have high mutation rates, and in the lab you can work with them, and they have large population sizes. And they developed some experimental work which suggests that, yes, viruses can behave as a quasi-species. And then there were a number of years of vigorous and acrimonious debate on whether this was true or not, which I think most people are tired of at this point. And I guess and so in viral circles, quasi-species, I think the critics of quasi-species would say, well, yes, viruses have high mutation, RNA viruses in particular have high mutation rates, and they exist in large population sizes, and there's diversity, genetic diversity there, so therefore they are a quasi-species, and it's almost become, in the literature, a term that means genetic diversity, where you have more than one clone. People even talk about bacterial quasi-species, where you get multiple different bacterial genotypes in an individual sample. And that's probably not the best way to use the term, because the term actually draws from a theory that makes certain predictions, right? And so, for example, in human viral infections, yes, you can find a lot of diversity, but the population isn't infinite, and it's not near infinite. There's not, you have a lot of bottlenecks and things going on. And so for that and other reasons, it's not clear that in vivo, these populations are actually behaving like a theoretical quasi-species. And so, applying that sort of model may or may not be the right thing when you use the term. And so I've just avoided it. One, because I'm starting my career and I don't want to make any enemies. But I think it's a term that's kind of used a lot, but it might have outlived its utility.
Is there something else you use?
I use a genetically diverse population, or a population of viruses. Sometimes I'll use a swarm.
Right.
Mixed population. I feel like it's better just to describe what it is as opposed to a term which might mean different things to different people.
Okay. So the key is that if this were a stock of polio, right, there would not be one sequence in this.
There would be
how many?
Lots.
So if there were a million,
that would be a lot of polio.
Well, let's say if there were a million infectious units, would there be a million different sequences?
There would be, I don't know if there would be a million, but there would be thousands of different sequences.
So are we technically able to sequence every genome in this stock and answer that question?
Yes, but there are limitations depending on how you do your sequencing, which is something we're very interested in currently in the lab. Is there's all these different ways of sequencing, a lot of next generation sequencing platforms, and because people want to understand the diversity. To understand the diversity, you first got to catalog it and define it. That's a problem we're interested in. And it becomes very tricky very quickly because a lot of these mutations that you're trying to detect are very rare because most mutations are bad. And so the virus will make a mutation, but often that genome is a dead end. It can't replicate anymore, it can't do anything, or it's a very unfit genome. And if you have an unfit genome, it stays at a low frequency and is lost, as opposed to increasing in frequency in a population. And so if you're trying to find things that are 1 in 1,000, 1 in 10,000, that's very tricky. And you get into a lot of signal to noise problems. And then you get into problems of, to look in that fine level, you always need to do a reverse transcription and PCR. And so, reverse transcriptase has an error rate, and so you get into issues of, is the error due to reverse transcription, or is it due to the viral polymerase? And there have been interesting approaches to try to tease that out. But it's a problem that you think, you can just do it, right? You can get billions of base pairs of sequence now. But then knowing which base pairs of sequence to believe and which ones not to believe are tricky.
So this is a technical issue that nonetheless, people are interested in solving, so you can understand what
Yeah.
constitutes a virus population.
Right. And then, one thing we're interested in is, well, if something is that rare, is it important, right? And or, trying to get virologists not just to talk about diversity as, total number of mutations, but how frequent are the mutations? Because a mutation that's in the population at 1% is probably different or playing a different role than something that's at 0.1%,
Yeah, right.
because the frequency actually probably makes a big deal.
Now, if I take my polio and grow it in HeLa cells, is this population of mutants going to be different? I couldn't call them mutants, just variants, I guess, right? Is it going to be different from poliovirus that comes out of someone's intestines?
For sure.
Can we see that right now?
I can't cite you the papers, but I'm sure. Yeah.
Yeah, I don't know if it's actually been done, but technically we can do that experiment.
Yeah, we've got lots of HeLa cell polioviruses that we've sequenced, and people have sequenced out of intestines. So the data's out there.
Because
and I would be highly surprised if they were similar.
The general point is that if you put a virus in different selections, HeLa cells versus the gut, you're going to have a different variant population,
Exactly.
right?
Exactly.
And that's something you're interested in, correct?
That's in essence. I should quote you and just kind of put that in my significant statement all the time, because that is the core of the issue. It's like you can find diversity, but what does it mean, and what does it mean in different scenarios, and can we start understanding this?
So that is what you're interested in. So tell us how you're looking at that.
Yeah, so we're doing a few different things, probably too many different things. I've continued the polio work that I was doing as a postdoc, and then we started working in flu since I came here. So one thing we've done, work I started as a postdoc, is like, well, if you want to know diversity, it'd be nice to be able to manipulate it, right? Have some control as opposed to just taking your HeLa cell stock and whatever you get, you get. You need some way of, can you manipulate this swarm to be two different things? And I had this idea as a postdoc to explore this using this concept of synonymous mutation. And so I know you've discussed on TWiV these codon-altered viruses that Eckhard Wimmer pioneered, and a number of other people have worked with. And we got some of the viruses from Wimmer because I thought, well, gee, the genetic code is degenerate, right? So you have, each amino acid can be encoded by multiple codons. And so, for example, the extreme one I use is arginine, which has 6 codons that code for it. But so the protein has always got an arginine at that position, and so it should be the same in terms of its function. But if you look at the connectivity of the genetic code, different codons mutate to different codons. So they tend to mutate either non-synonymously or synonymously or non-conservatively to a different degree. And so the virus starts at the same place in terms of it makes the same amino acids in the same protein. But if you do a lot of synonymous mutations, you're actually, when the virus mutates, which polio does at a high rate, it's going to generate different variants. And so you're going to have a different population. And so we did this with Wimmer's viruses and showed that that actually made a huge difference, and that you could get viruses that had different fitness in cell culture, had different sensitivity to mutation because maybe they're throwing the wrong mutations at the wrong place, and actually were different in mice. And so we're continuing that work a bit. We just published a paper where we had kind of an algorithm or an informatic approach to design more of these viruses, because it was a neat trick. We got it with 2 viruses, but there's a lot of controls that we would like to do to incorporate, and so we developed a tool to generate viruses that have a large number of synonymous mutations, but control for other features. So we might start exploring some of these viruses, because you can synthesize a poliovirus at reasonable cost now. And then we're also looking at kind of mutational tolerance and how that affects the viral population. And then in the flu work, we've done a little bit of that as well, but One thing we're interested in and very excited about is we've teamed up with some fantastic epidemiologists at the School of Public Health who run studies of vaccines, also look at have household cohorts. So we're interested in looking at flu transmission in nature, in people. And so we've developed tools to sequence these populations, understand the diversity of these populations in the setting of vaccination or no vaccination, and also understanding how much of the population goes from one person to another in a kind of a natural setting. And so that we spent a lot of time kind of getting our ducks in a row and our protocols fine-tuned, and we're just starting to get data back on that, so it's pretty exciting.
So you think that you will eventually be an influenza lab after polio is eradicated?
Yeah, I don't know. I think so. I think flu's fun. I just like questions, and so I tell people I don't consider myself a polio virologist or a flu virologist. I'm just a virologist who's interested in evolution. And so, for now, I like the polio stuff. I like the flu stuff, but I've already got some other viruses that we might want to, start looking at. I think the one thing I like about the flu work is because we've got these great collaborators, we can actually look at, viruses infecting people and actually, understand it in its kind of natural context. And so I'd like to do more in that area. And so I'm not really, I don't want to limit myself by system.
Okay, fair enough. Could you make a flu vaccine based on the recoding Wimmer strategy?
People have. I mean, not to market, but I think Martinez Sabrito had a paper where they recoded NS1. People have done it, as you guys discussed, with VSV and dengue and chikungunya, and there's been a number of papers.
There's quite a controversy about what is the actual mechanism.
Yeah.
What is it? Codon pair bias versus something else, which I forgot. We'll have to have you come on TWiV sometime and sort it all out.
Yeah, I mean that was one of the motivations for this paper that we just published is you, by making synonymous mutation, you affect a lot of things. Potentially RNA structure, potentially codon bias, potentially codon pair bias, CPG content, dinucleotide bias. There's a lot of things you're doing. And so we're like, well, gee, is there a way we can do this and control for all that? Can you control and make sure that all these other metrics are similar? And you can. And so we, a postdoc in my lab, Daniel, published this paper and essentially using all these metrics and trying to control for all of these things as best we can. So hopefully that'll help solve this problem.
So one last thing, and we'll move on to Akira. You just published this influenza paper showing that you can push the virus over the error threshold with a mutagen, right? So this is something that we've thought for some time, that RNA viruses exist at a level beyond which it's very close to pushing them into lethality, right? So you've shown that with flu, right? But in a paper by your former postdoc advisor, he's using circ-seq to try and get over the errors in sequencing to define a population. He says maybe they're not so close to the error threshold after all. I don't know if you're aware of that or
I mean,
have any thoughts.
So what we do know is that, I mean, we didn't come up with the idea. We just applied it to flu. I mean, John Holland, years ago showed with VSV that with a mutagen, you could force the virus to make too many mutations. I think what we don't know is how close some viruses are to that threshold than others, and probably different viruses differ in terms of, and that's why we're interested in mutational tolerance, because some viruses might be more tolerant of mutation than others. And so a little bit of comparative virology might help.
We do tend to make broad conclusions based on one virus, right?
Right.
So the error threshold was originally polio-based?
The error threshold as a concept dates back to Eigen and these theoretical studies. And I'll probably get a horrible email from James Bull if I don't clarify this, is that the error threshold and lethal mutagenesis are sort of different. And so lethal mutagenesis, the error threshold is kind of this melting of the population into loss of information. And so you can just tell that's kind of a theoretical concept, whereas lethal mutagenesis is you increase the mutation rate, and so it's almost like counting, like you know more of the viruses are going to have a mutation that's lethal, and once you get enough of those, there's not going to be any infectious virus left. And so how well that works depends on the mutation rate of the virus and then how damaging any given mutation is. And with the flu, it's interesting because we didn't publish this, but favipiravir, which is a flu drug that is, I think, approved in Japan for use, and I think is undergoing late-stage trials here. A group actually showed that it actually functions as a lethal mutagen. And then I think since it's also been shown to be lethal mutagen for norovirus. So we're starting to see, you know, it used to be something people would just did in the lab, but now we're seeing an actual drugs that are going into people.
It's clear, though, that for DNA viruses, you have to add a lot of mutations to the genome before you go over this threshold, right? Before you, I should say, lethal mutagenesis occurs, right?
Yeah, you know, I don't know the data well. I mean, I think people have done it in DNA viruses. Yeah. I think the trick is, I mean, probably one reason is RNA viruses, it might not be just because they have high mutation rates, but it might be because how damaging mutations are, how tolerant they are of mutations, because RNA viruses tend to be small, have kind of compact genomes, overlapping open reading frames, so it's easy to mess that up. Whereas DNA viruses are like SUVs, and they just, you know, you can do something to them and they just don't care.
DNA viruses are like SUVs. There could be a title.
Could be.
I mean, large, you know, not the small DNA.
Got it. So
It's more like a mini.
those are sports cars.
have you seen this latest paper on single cell?
It's in the virtual stack on my computer.
All right, so we're going to do this on TWiV in a couple of weeks when everybody's all together. Maybe you want to join us.
I mean, they do great. That lab does great work.
You can explain it to us.
Yeah, be fun.
Right. Okay. Akira.
Yes.
You're interested in HIV and flu also?
It just started, yeah.
So I actually read a couple of your papers last night. I had Akira take me home early so I could read some of your work. And I read a couple of really cool stuff that has to do with HIV assembly. But I'm going to let you tell me what is exciting that you'd like to talk about.
Right. So our lab is mainly interested in how viruses, enveloped viruses, use the plasma membrane for its assembly. And once it assembles, how that assembly at the plasma membrane affects the next process, which is to spread to the next target cells. So the two main aspects are for HIV, we are looking at how HIV determines the virus assembly at the plasma membrane, and the driver is the protein called GAG, which is a building block of retroviruses, including HIV. And this guy has some region at the N-terminal, amino terminal side that has lots of basic residues. And the people have been saying that these basic residues are important for binding to the membrane because membranes contain acidic lipids, the basic acidic interaction. But the HIV seemed to be specifically going to the plasma membrane, not any other membranes. Any other membranes also have acidic lipids, so what's the reason for this specificity? So there is one lipid specifically localized at the plasma membrane called phosphatidylinositol 4,5-bisphosphate, or let's say PIP2. And this PIP2 is acidic lipid specific to the plasma membrane, and when we reduce the amount of this, then HIV assembly takes place in the other place, or GAG protein fail to bind the membrane, so there's no virus particles formed, either fate. So that means that this lipid is important, and basic residue, we as well as other group has also shown that this interaction of these basic residues and the lipid head group is important for localization of GAG to the plasma membrane, and then assembly at the plasma membrane. And then that sounds like a straightforward story. One protein and one lipid binds, and then one lipid staying at the plasma membrane, so it got to be at the plasma membrane. And that is in sort of a parallel to the many cellular proteins that use PIP2 for localization to the plasma membrane. But there was a twist, and the twist was that this basic residues in GAG also binds RNA. And so there must be some kind of competition. And it turns out that RNA is actually blocking these basic residues from binding to other acidic lipids. PIP2 can override this RNA block, but the other acidic lipids, such as phosphatidylserine, cannot override this. So that we proposed is the mechanism to specifically bring this HIV GAG protein to the plasma membrane. We try to find what this RNA is. So what we did is in vitro experiments, strip off the RNA from GAG first, then add back different RNAs. As a first test, we use yeast tRNA, which is a generic lab material. So let's see what happens. And then it does inhibit the membrane binding. Oh, so any RNA could work. And then, okay, well, that's good to say. But then it turns out that other groups showed that in cells, actually this basic region specifically binds to tRNA. So we are right, but we thought it's a non-specific, but it's actually specifically binding to tRNA. So that's an interesting thing. And then question arises is that why is GAG doing this convoluted method to go to the plasma membrane? Why not just binding to PIP2 is good enough? We thought that maybe RNA is coming first. And then HIV found that, we could use this PIP2 thing to go to the plasma membrane. And it may be, the evidence that we found consistent with that idea. And that idea is that, so we compare the HIV GAG protein and the GAG protein of other retroviruses, especially HTLV, human T lymphotropic virus. HTLV has a basic sequence, but it's scattered around. So small basic patches all over the place. HIV has a big basic patch. This HTLV isn't really inhibited by RNA, and it doesn't require PIP2, but it goes to everywhere. So it doesn't have a specificity just because it binds to any membrane and not restricted by RNA. But when we introduce into this HTLV more basic residues, maybe sort of gain of function. But then this guy is now inhibited by RNA. So we think currently at least tRNA or any RNA maybe binds to a large basic patch found in the retroviral GAG proteins. And the GAG protein of HTLV probably made the basic patch small enough and then stay small so that it's not going to be inhibited by RNA. It binds to everywhere, but anyway it gets out. Whereas in case of HIV, it got the large basic patch, but that gets suppressed by RNA. So HIV had to find something else to bind the membrane, and then it turns out PIP2 is a useful tool for overriding this RNA block.
So PP2 binding is not inhibited by RNA?
Right. So
tRNA.
you don't need to. Yes, that's it.
And the tRNA is to prevent a GAG from binding to other lipids elsewhere in the cell that would distract
Right.
it from its plasma membrane destination. Is that the idea?
Yeah. So the tRNA is able to inhibit the binding of GAG to, for example, phosphatidylserine, which is the most abundant acidic lipid in the cell. So that's how, we think it's going on.
Do these tRNA? I know tRNA is bound to the genome when it gets packaged, but do these other GAG binding tRNAs get into the particle?
So there is not much tRNA incorporated into the particles, and then that is consistent with the idea because once GAG binds to the membrane, those tRNA bound to this basic region will fall off from the GAG. I mean, that's a model.
And this is for HIV, and you say HTLV is different. What about other retroviruses?
Right. So in our system where we make only this N-terminal domains varied and then the downstream region all same as HIV, we found HIV and Ralph sarcoma virus GAG protein are similar in that it's suppressed by RNA and require PIP2, whereas murine leukemia virus and HIV are the other group.
Okay. We don't understand why there's different ways to achieve this.
Yeah, that's why question is always difficult.
You're right. You got me. You got me. Let's put it another way. What's the function of having this diversity? See, I didn't say why.
Okay.
Just whatever works, I guess.
Right. Yeah. I guess we are still in the halfway for this entire thing. I mean, without knowing the entire thing, how can I say halfway? I don't know. But the thing that we are using is a tissue culture system using HeLa cells, which is not the actual natural host for the viruses. So we are moving toward the T cells to make sure that this is all the picture or something else remaining.
So you said that cell proteins also bind PP2?
Yeah.
Do they go into the particle as well?
Some of them do, but the incorporation of the proteins into the particle is another thing that we are interested. So what kind of cellular proteins gets into the virus particles, and retrovirus particles incorporate lots of stuff, and that is partly because it binds to the plasma membrane. And what we found before is that the binding of GAG protein to the cytoplasmic face of the plasma membrane induces the reorganization of the plasma membrane microdomains. And so incorporation is not passive process, or not passive for everything, all the proteins. And I guess there are many. Well, there are at least a few host restriction factors that is making people heat it up. These are, for example, tethering, and also recently serine-5. And so they may be incorporated more specifically because of this reorganization of the plasma membrane. Yeah.
Are you thinking that there might be some cell proteins that would be beneficial for the virus as well?
Yeah. So that's something we are interested. So the another part of what we are interested is that where on the plasma membrane virus particles are assembling, and not just how it gets to the plasma membrane. And several years ago, we found that HIV GAG proteins localized to the tail end of the migrating lymphocyte called uropod. And we showed that this uropod is a precursor or a constituent of the virological synapse where HIV hops to the next target cells. But even when T cells are not polarized, T cells are round, then these uropod proteins and GAG proteins are co-localizing quite well. And then that co-localization appears to be a specific process because without GAG expression, these uropod proteins, some of them, let's say PSGL1 and ICAM3, for example.
Right.
Both of them are uropod protein, and then they co-localize. But when we express GAG take the PSGL1, but get rid of ICAM3. So there must be some reason for this. We are looking for the reason right now, and there may be a possible chance that some of these uropod proteins are actually helping the virus spreading. Yeah.
Okay. All right, one more question for you. So every cell has endogenous retroviruses, and they make proteins, they make GAG.
Right.
Do they interfere, participate in this process at all?
Right. Yeah, so the classic case is that FE1, so the incoming MLV would be inhibited by these endogenous retrovirus fragment of GAG. But we also found something similar in case of HIV. So we in the human genome, there are lots of endogenous retrovirus, and some of them are totally dead virus and no longer making any proteins. But there is a group of the endogenous retrovirus HAV-K, and when we express GAG protein of this HAV-K, then what we saw is that it co-assembles with HIV GAG at the virus assembly site, and that suppressed the HIV release efficiency, and also the virions that gets released from this cells that express HAV-K GAG has a reduced infectivity. So theoretically, it is possible that HAV-K GAG, which is already present in the human genome, can be the sort of restriction mechanism against the retrovirus similar to HIV. Why I say theoretically? Because it's all in tissue culture system, and we haven't really. It
would have to be in the right cells also.
Right, that's true. Yeah. But the David Markovit's group here at the University of Michigan shows that HIV and the UCSF group, I'm blanked about the name, show that HIV infection can increase the HAV-K expression.
Doug Nixon, I think.
Yeah, I guess that's right. Yeah.
Yeah, you should know, right?
That's right.
All right, Kathy, it is Friday. We're doing a TWiV. Time to do some picks of the week.
So I picked something that's a theme that we've talked about on TWiV a lot, and that's women in science. And it also relates to another thing that we've had, which is Lego models that are women in science. And so October 13th this year, the second Tuesday of October, is Ada Lovelace Day. And it's celebrated around the world. People do seminars and things to try and increase participation and awareness of women in science. And Ada Lovelace's 200th birthday will be this December 10th, 2015. And in recognition of that, someone has created a potential Lego model, which is Ada Lovelace and Charles Babbage. So who is Ada Lovelace? She's widely credited as the first, she's a mathematician living in the 1800s. She died actually at a very young age. She lived from 1818 to 1852. She's widely viewed as the first computer scientist. And so in conjunction with Charles Babbage, he built or designed a model for the first general purpose computer, and she wrote the first program for it. But she didn't get to execute the program because he never got to build this computer. But the models are here, and I'll show you guys, and maybe you'll just all check it out. I'm happy to send the link to you if you want it before Sunday when this TWiV comes out. But it's really cool because evidently inside of this computer model, there's space to have a Raspberry Pi computer, which is a single, I didn't know exactly what it was, and my brother was appalled, the computer software brother. But I'm sure Pat knows what Raspberry Pi is, and they're going, oh yeah, of course, you should have known that. But Raspberry Pi fits inside of this, so that's pretty cool. And what I liked about it is that how they made Ada's dress to be long. It's just this angled back thing, so it's pretty cool. Anyway, those of you who've listened to TWiV know that you can go to the LEGO site, you can look at this, you can vote for it, and the more votes it gets, the more consideration LEGO will give to building it as a model. And they never send you spam. I can vouch for that. You just go register and you can vote for this.
So this is a model of what he had designed but never built, right?
Correct.
So I think that, and they have in your picture, they have Babbage and Lovelace standing there, and she's very severe with this long black dress, and her eyes are extremely angry, and he's like, I'm sorry, I never built it. I'm reading that into it anyway.
Right.
So this is really cool. All right. My pick is something from here at the University of Michigan. It's a blog called My Sci-Writers. It's some science writing by a couple of people here at the university. I found out about this yesterday when I talked to the person who has developed this, Ada Hagen, here at University of Michigan, and she's interested in science writing. Is Ada here? That's probably. Yep. Yes, hello. And she's gotten a few PhD students together to write about science in a nice way that informs the public. So we'd like to give it a TWiV bump, and that's my pick of the week. My Sci-Writers. Some good articles there, so check that out. We have 2 listener picks. Junio has been following our podcast for years. He works at Google. He's an IT guy. It's really cool. He sent a link in honor of World Polio Day. Did you know that Bill Gates writes a blog? Supposedly, right? But Bill Gates has a blog, and today's post is a XKCD comic that was made for World Polio Day, which is pretty funny. So check that out. And then we have a listener pick from Carrie, who's here in the audience. She writes, I should have thought about this earlier this week, but it just occurred to me this morning. There is a fabric website called Spoonflower where you can design your own fabric and then have it printed. You can also order fabric that other people design. They do a themed weekly design contest, and this week it was microorganisms. While my design did not win, they have the top 10 posted on their website, and you can search for more specific designs. I've designed and ordered fabric there for some fun sewing projects for virologist friends, and I bet there are other microbiologists out there that would love to do some microbe-themed sewing.
I can verify that Kari made skirts for my 2 little girls and a T-shirt for my son that have virostemes and are very.
And Kari is a postdoc in your laboratory. Yeah. I'm sorry where is she? Here. I'm sorry your design didn't win. Can you send it to us so we can check it out?
Okay, these are really cool.
Yeah, they're very neat.
Very neat.
You can make all sorts of things with this.
Spoonflower?
Spoonflower.com.
Which kind of flower?
Like a.
What's the name of it?
It's the name of the site.
F-L-O-W-E-R.
Spoonflower.
F-L-O-W-E-R.
Yeah.
And this, so I did an interview with Michelle Banks, who's a microbial artist, and she makes scarves. So this would be a perfect way to make your own scarf as well. So thank you for that, Carrie. It's Kari, right? And I have to apologize to Harry. I pronounced your name wrong all day yesterday on TWiV.
Used to.
I'm sorry. Me too. Believe me. Mobley, right? Harry Mobley.
Hari.
Oh my gosh. Were you going to say something? Sorry.
Yeah, well, I was just to say that there is a retrovirologist who has a retrovirus patterned kimono in Japan.
Really?
Yeah.
That's nice.
Yeah, it is. Yeah.
That's really cool. All the things we learn about microbiology. All right, this episode of TWiV will be found at iTunes and also at twiv.tv. And if you have an iOS or Android device, there are apps that you can use to download it for free and listen to learn all about microbiology. I want to thank our guests today,
all at the University of Michigan. Mike Imperiale, thank you so much for joining us.
It's been great.
Adam Loring, thank you.
Thank you.
And Akira Ono, thank you very much.
Thanks for having
Could
me.
you count to 6 for me in Japanese?
1, 2, 3, 4, 5, 6.
Roku. And
Yeah.
of course, Roku is the 6th company started by the guy who makes the Roku device, which I really like.
Okay.
It's a free plug for Roku. Kathy Spindler is here, also at the University of Michigan. Thank you, Kathy.
Thanks, Vincent. This was really fun.
And thanks to the Department of Microbiology and Immunology and Harry Mobley for bringing me out here and allowing us to do 2 podcasts. Thank you. Thank you. Harry never says you're welcome. Did you notice? And thanks to the audience for coming. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.